In this study, we are interested to discover what are the microbes in the gut microbiome of individuals with multiple sclerosis are doing. For example, what chemicals are they producing? So in this regard, we assess the association between multiple sclerosis and the functions of the gut microbiome by comparing MS cases to unaffected controls. I'll briefly summarize the methods and the results. All these details are available in my poster. Participants in the study were enrolled if they were 21 years old or less and had symptom onset at the age of 18 or less. We have three major aims in the study: to assess the diversity of enzymes and proteins, to assess the relative abundance and prevalence of metabolic pathways and gene groups, also known as gene ontologies. In both of these aims, we compare MS cases to unaffected controls. Then we performed a subgroup analysis to assess changes in the results by disease-modifying drug exposure, also known as DMD. We used shotgun metagenomics to sequence the gut microbiome. All of the cohort characteristics were similar, including stool consistency. We assessed alpha diversity, which is the diversity within the stool sample. There were no differences in the alpha diversity, enzymes, and proteins by disease status or DMD exposure. We observed differences in pathway abundance, such as a depletion in homolactic fermentation pathway, which produces lactate. Interestingly, peptidoglycan maturation is higher in MS cases exposed to DMD versus MS cases naive to DMD exposure. There were also differences in pathway of prevalence, meaning presence versus absence of pathways. Methanogenesis was more prevalent in MS. There were differences in gene ontologies by relative abundance and prevalence. Again, we see the association with MS and methanogenesis. In summary, while there were no overall differences in the diversity of biological functions in MS cases versus controls, we did observe associations between microbial functions with MS. What stood out to me the most when comparing either pathways or gene ontologies was that genes related to methane production from archaea are more present in MS versus controls. Methane can cause constipation or make constipation worse. This was a large collaborative study. I want to acknowledge my supervisor, Helen Tremlett, my co-supervisor, William Shao, many collaborators, and my funders from the MS Society. Thank you.